So the rest of the course is talking about infection of people and some animals, how viruses cause disease. So you're ready for this. I'm going to talk about mechanisms of pathogenesis, immunity, and some specific virus infections. So today we'll start with some basics about infections in general. And of course you have next week off to think about it. And then we'll resume when you get back. So the rest of the course we're going to talk about host-parasite interactions, infections in the real world. So far we've talked mostly about infections in cell culture. We're going to move into the real world. And it's really, we're going to focus on part 3 of this strategy that we talked about early on. All viral genomes are able to establish themselves in a host population so that virus survival is ensured. It probably sounds a little amorphous to you, but as we do some specific virus examples, you understand what that means. And I suggest you have a look at Appendix B in Volume 2. This is pathogenicity snapshots, a little summary of all the important viral diseases that we're going to talk about. The viruses involved, epidemiology, disease mechanisms. So some basic facts. Every successful virus that is with us today has to modulate host defenses. As you'll see in a few other talks, the host mounts a great defense against virus infection, and without counter defenses, viruses would be eliminated. And every host has intrinsic defenses and immune defenses that are evolved to deal with both infections and tumors. We'll talk a little bit about tumors as they are caused by viruses. But the immune response also deals with other kinds of tumors as well. And the interface between the host and the virus at this level, the offense versus defense, really is the front line. What happens very early on determines what will happen through the course of the infection. And as I told you, the very first day, we live and prosper in a literal cloud of viruses. Most infections have no consequence. Sometimes the particle never finds the right cell to infect, and you already know somewhat what a virus needs in order to infect. So many viruses land on your skin, and as we'll see, that's not a very good place to start an infection. Many viruses are destroyed or inactivated as they enter the host, and many infections never go farther than 1 or 2 cells. A lot of viruses enter your respiratory tract and may grow in a few cells, and then they're cleared for one reason or other. We'll talk about the mechanisms that are involved in that clearance. Even if we do get infected, if a virus can replicate in many cells, many infections are what we call inapparent. I'll use that word a lot. An inapparent or subclinical or asymptomatic infection. There are no symptoms. The virus's replicating immune defenses are activated. Sometimes antibodies are made. And in fact, that's how we know that there are so many inapparent infections. We look in people and we find antibodies to so many viruses. 80% of the population has antibodies to certain polyoma viruses, and we don't remember ever having any illness because it's an inapparent infection. And that perhaps represents the most advanced symbiosis between the virus and the host. The virus is replicating and surviving in the population, and the host survives as well. Of course, the virus doesn't care if you get sick and feel badly. It just wants to transmit from one host to another. So there's maybe not a lot of distinction between inapparent and very mild infections, but we'll talk about that later. And viruses are, as I said, viruses are often replicating. So here's a great example of a inapparent infection, West Nile virus. At the end of this course, we'll have a guest lecture on West Nile by Professor Despommier, who wrote a book called West Nile Story. And he'll tell you all about how this virus spread to the U.S. in 2000. By 2004, there were about a million people in the U.S. infected, which we determined by antibody surveys. That's how you find out if someone is infected or not. And it turned out that illness accompanied with fever, febrile illness, developed in only about 20% of the infected people. So 80% of the people were infected, but they were fine. 1% of these people had neuroinvasive illness, encephalitis typically. After West Nile, it invades the brain and replicates in the brain. And that's the most serious consequence of a West Nile infection. But you see, it's very small. So many people were infected with no obvious disease. That has its own consequences. If you don't know that there's a virus present, then you take blood from people. We have to have a blood supply which is based on giving blood still. You can transmit virus infections if people don't know that they're infected. So before you know to screen the blood supply for a particular virus, it can be spread. And also, if you have a lot of people infected without symptoms, it's hard to stop an epidemic. Great example, polio in the 1930s and 1940s in this country. One of the measures that was taken to stop polio in places like New York City, where it was rampant, was to quarantine the people who were paralyzed. Put them in places where they couldn't contact. It didn't do anything to stop the disease. Anybody know what quarantine means, by the way? What does it mean?
That you like isolate the people.
Oh, yes. You're isolated. But where does the word come from? Sorry. Quarantine. 40 days, yes. Keeping away for 40 days, 40. So for polio, it turns out that only 1% of the infected people are paralyzed. So 99% of people are shedding virus. They're quite fine and walking around, and they're spreading infection. So quarantine is useless. And I would be so bold as to suggest that quarantine is never useful. We were just talking about this at lunch. We had a flu expert give a seminar, and he was saying how at the peak of the pandemic last year, airplanes flying into China, they would check everyone for fever. And if you had a fever, you would be put in a hotel for 2 weeks. This is useless. Because in 1957, before there was a lot of global air travel, the pandemic strain that emerged that year traveled around the globe in about 6 months. So the virus is very good at traveling person to person. It doesn't need airplanes or trains or boats. So quarantine is pretty bad and useless. But and this is one reason why, because there's so many people who are infected without knowing it. Now, one day, we're going to have a tricorder. Do you know what a tricorder is? It's a little thing. It's about this big. And if someone is sick, you just wave it over them, and it tells you what virus or bacterial infection they have. We're going to have that, I don't know how many years from now. And then we'll know, everyone, as you walk in this door, there'll be a sign up here. No, it'd be better yet. I'll get you in 1 second. You're going to get up in the morning and look in the mirror, and there's going to be a sensor in the mirror that says, oh, clear. Yes, sorry.
With the not exhibiting any symptoms of disease, is that just because it hasn't had time to accumulate enough for you to exhibit symptoms, or is it just that it will never exhibit symptoms?
No, there is actually plenty of virus in these people who don't have symptoms. That's not the issue. It is just that it hasn't triggered. As you'll see in a few lectures, many symptoms of disease is often a robust immune response. And the virus simply hasn't done that. It has evolved to be silent. That's probably part of the strategy for spreading in a population so that you're not detected. But yeah, you have full-blown infections for most of these inapparent infections yet. And in many cases, we don't understand why. But it's not because the virus isn't growing well. Okay, 2 facts and an apparent paradox. All hosts have remarkable defenses, as I've been telling you. Yet, in some hosts, infections proceed. We get sick and we die of viral infections. Why is that? What happened to the defense system? How can viral genomes, which often encode only one gene, how can they do this? How can they make us very sick and kill us? Well, that's the whole topic of the second half of this course. And we don't often know why. Here's another example, which I learned today. So there is a very highly pathogenic strain of avian influenza virus, which you may have heard of, H5N1 avian flu. Anybody hear of that? So, you know, it kills birds on poultry farms, and occasionally it infects people. It's infected about 400 people globally, but it hasn't spread yet from person to person. There are about 3 documented examples of spread from person to person. They all occur within families, and all occur within the bloodline. In other words, mother or father and children, it spreads among them, but never husband and wife. So that tells us there's this genetic component to being susceptible to transmission with this virus. We don't know what it is. And eventually one might be able to find out with enough data. But that's what probably determines whether you get ill or not. You have some suboptimal immune response of some sort that's genetically determined. And for the most part, we don't know how that works. There are a few examples that we'll get into, though, in this course. So these are some of the fundamental questions about how viruses cause disease. How does the virus get in? What's the host response? Where does the virus first replicate? How does the infection spread? What tissues are infected? Is the infection cleared? Or is there a persistent infection established, which means the virus infection goes on for years? And how is it transmitted to other hosts? And these are some of the, we'll try and answer all of these questions in the second half of this course. There are 3 requirements to get an infection started in us or any animal host. You have to have enough virus particles to start the infection. And wherever the virus lands on you, and we'll talk about where it can land, the cells must be susceptible and permissive and accessible, of course. And initially, the antiviral defense systems have to be either absent or defective in order to allow the virus to proceed from that point. So let's just briefly talk about this issue, the sufficient virions at the site of infection. So this is a question everyone would like to know the answer to. How many virions does it take to infect a host? And it's almost impossible to answer this because in the real world, for most viruses, we can't actually do the experiment and infect people. For most viruses, that's not ethical. You can do it with a few viruses that cause very mild infections, like rhinoviruses. So we know that a few 100,000 rhinoviruses, when put into someone's nose directly with a dropper, is enough to start a cold. But that's also artificial because it is not an aerosol, which is normally how you would get a respiratory infection, or touching a hand and putting it in your nose. So it's a very difficult question to answer. And it varies for every virus and host interaction. But here are some of the variables that make it difficult to answer this question. First of all, genetics of the host. Everyone is different in terms of their susceptibility, and it's controlled by specific genes. We're an outbred population, so there are a lot of different alleles for every susceptibility gene in the population, and a lot of that is difficult to sort out. The host defenses can intervene to make it more or less difficult to infect with a certain number. The character of the virus and its ability to cause disease is an issue. Host social behavior. Different kinds of interactions among hosts promote infections in different ways. The age of the host also makes a difference. Weather, environment, nutrition. So you see there are many factors that will affect how much virus it requires to infect you. The natural environment that we live in is a considerable barrier to infection. So the viruses floating in this room, for the most part, don't have much of a chance to infect us for a variety of reasons. They're little guys, and they depend on air currents to push them around. Once these viruses are expelled from us, they're diluted tremendously in the air and in the environment, so the MOI goes down, and that reduces the chance of infection. Sunlight, drying, the environment in general, they all reduce the numbers of infectious virions. You know, a swimming pool with lots of chlorine is not a hospitable place for many viruses. So there are a lot of obstacles to a virus infecting you. But interestingly, viruses have evolved to overcome some of them, which is why they can infect us. So although many particles are sensitive to heat and drying and UV light, the way to get around this is to make billions and billions of progeny. And then at least a few will infect you. If you only made 1,000, you'd have no chance. But the replication cycle has evolved to produce hundreds of thousands of particles per cell. That's why it has, one reason why it's done that, so that it can overcome these environmental obstacles. A lot of viruses are stable at low pH or resistant to proteases or even want proteases. These are some of the environmental variables that can inactivate them. So viruses evolved to become stable. And it's amazing that even enveloped viruses can become stable to proteases in low pH. Some viruses can survive in the gut, which is very inhospitable, and there are many that are transmitted by the fecal-oral route. And many virions never experience this wonderful environment. They are spread by insect vectors, so they replicate in your blood, and a mosquito takes a blood meal, takes some virus up, flies away, and the virus is carried by this nice mosquito vector, so it's protected, and then it infects another host, so it never sees sunlight, UV, low pH, etc. You can imagine many other examples of that as well. Yes?
Had any specific viruses developed the ability to, let's say, be immune to UV, or it's just that they aren't exposed to it because of some pain?
Not completely, but to be quite resistant. Can you name one example of a virus that's quite resistant to UV inactivation? We talked about it. Yeah, the retros, because they have 2 genomes, right? So they're very resistant compared to most other viruses, but none are completely resistant. But you know, it's all a matter of degree. It's not an all or none. Viruses are not binary, as I like to tell people. And many infections are spread by physical contact, so the virus doesn't have to be floating around in the environment. Body fluids, for example. So let's talk about how viruses get in, gaining access. And the site of entry is very important here. And the human body only has a limited spectrum of entry sites for viruses. And they're shown here. There are 5 different ones. The eyes, the conjunctiva of the eyes, the skin, the respiratory, alimentary, and the urogenital tracts are really the ways viruses get in. The skin, let's talk about first. Biggest organ in the body. Unless it's broken in some way, it's pretty impervious to infection. It's a great barrier. The outer layer, of course, is dead. And if a virus lands on it, it will not replicate. But remember, viruses require a living cell in which to replicate. And in addition, your skin has other defenses besides the cells being dead. It's not terribly moist. It's have low pH. And there are many microorganisms, actually, that we've recently discovered that live on your skin that secrete products that inhibit virus multiplication. So these are commensals that are happy to live on us. We provide them with an environment in which to grow bacteria, for example. And in turn, they protect us against other organisms. So the skin is acidic. Its pH is about 5.5, and a lot of viruses can't handle that. So that's a diagram of the skin there. The outer layer, the epidermis, is dead keratinized cells which can't support viral infection. And the only way that you can get a virus through is to have a skin cut, an abrasion, an insect bite, an animal bite, a needle puncture. And so, in fact, when viruses land on your skin, the best way to get them off is to wash it. So hand washing is really good. But you shouldn't wash too much because, of course, when you wash your hands too much, you remove the oils, your skin cracks. And when your skin cracks, then the viruses get in the cracks. So there's a happy medium, right? And with all the dispensers of sanitizer all over the place, you have to resist the temptation to use one every 10 minutes. This will just dry your skin out. The epidermis doesn't have any blood vessels in it either. To get spread from the skin, the viruses have to get below the epidermis, where there are living cells, blood vessels, and lymph glands, lymph vessels as well. So if a virus can get through the epidermis, by the means we talked about, it can get into underlying tissues, replicate, and spread very nicely. Now, in contrast to skin, the mucosal surfaces of our body are ripe for infection. And these are, of course, the respiratory tract, the alimentary tract. Their function is to exchange gases and nutrients, so they have to be permeable. They're a wet environment, so they're susceptible. They don't have a layer of dead skin. But they do have defenses. So again, it's a game between the virus and these mucosal surfaces. Respiratory tract is the most common way that viruses get into us because it has a huge absorptive area. And our ventilation rate is great, 6 liters a minute. There are 300 million alveoli, the terminal sacs in the lung, a lot of alveoli, and all that is available. It's a big surface area with a lot of, we're always accessing and sampling the air. So great way for pathogens to get in. There are lots of cells in the tract replicating, and there's easy access to the blood. Good way for viruses to get in. So typically we inhale viruses from droplets that have been produced by coughing or sneezing, or we introduce viruses on contaminated fluid into the tract. Now, in a healthy person, our tract is well defended, and it has to be because it's a weak point. So we make a lot of mucus. We make between 20 and 200 milliliters per day of mucus. Yes, you all do, in your nose and the respiratory tract. And that's important for defense. In your upper tract, the mucus traps particles, including viruses, and then it's brought to the back of your throat, and you swallow it, and they're inactivated in your stomach. And you swallow on the order of 30 times an hour, unless you're taking a test, and then you swallow more. So that's a good defense against microorganisms. And then mucus moves in the other direction. There's an escalator, the mucociliary escalator, we like to call it, which moves liquid from your lungs up. So the mucus in your lungs traps microorganisms, and then the cilia move it up a centimeter a minute, and then you swallow it. So you don't know any of this is happening, but it's an active process for moving things out of your tract. So, and there's also filtering of particles in your sinuses. There are immune cells in your tract. There are antibodies. So it's pretty well protected. And this is what it looks like. Here's a diagram of the whole tract, the upper tract. So we like to divide the tract into upper and lower in virology. And it is generally lined with an epithelial layer, which is the first site that viruses will encounter. It's covered with mucus. These are ciliated epithelial cells. The mucus is made by goblet cells. And beneath it is what's called a basement membrane. It's not really a membrane, but it's a layer of adhesion molecules that provides a barrier to the underlying tissues. And depending on where we are in the tract, viruses can infect pretty much anywhere. They can infect the upper tract and stay there, and they cause a variety of syndromes. We call this rhinitis or pharyngitis or laryngitis, depending on what level of the upper tract is infected. If we go down further into the lung, we have tracheitis, bronchitis, bronchiolitis, and bronchopneumonia, the very lowest parts of the respiratory tract. So there are viruses that can do both. They can infect the upper and lower tract, like influenza, and some can do just the upper tract. Okay. The other portal of entry is the alimentary tract, where you're always putting things into eating and drinking. We put lots of things in there, and it's great opportunity for viruses because it's designed to move and absorb food, so the viruses are always moving, so they can encounter the wall of the gut, which is what they need to do to bind to a cell. Good opportunities for virus-cell interactions. As you know, the tract is pretty hostile. The stomach has acid. The intestine's alkaline. There are all kinds of digestive enzymes and so forth, biodetergents, antibodies, and mucus. So that's the defense of the alimentary tract. But viruses can get around it. They have evolved to be resistant. You know, the early viruses that pass through a digestive tract, if a billion goes through and they're all destroyed, maybe one survives.